Segment 1. People have been experimenting with exogenous substances in order to change the functioning of their bodies and brains as far back as we can trace human history. Exogenous means arising from outside the body. The brain is an electrochemical system in which most drugs affect the nervous system by altering brain chemistry and synaptic transmission. Neurochemistry is the branch of neuroscience concerned with the fundamental chemical composition and processes of the nervous system. Neuropharmacology, also called psychopharmacology, is a scientific field concerned with the discovery and study of compounds that selectively affect the functioning of the nervous system. Synaptic transmission is a complex electrochemical process. Because the action potential strongly depolarizes the axon terminal, voltage-gated calcium channels in the terminal membrane open. The resultant influx of calcium ions drives the migration of synaptic vesicles to the presynaptic membrane where they release the neurotransmitter molecules into the synaptic cleft. This process is called exocytosis. Receptor, also known as a receptor molecule, is a protein that binds and reacts to molecules of a neurotransmitter or hormone. This mediates a response on the postsynaptic side. The transmitter molecule binds to the receptor, which changes its shape to open an ion channel or altering chemical reactions within the target cell. An ionotropic receptor is a receptor protein that includes an ion channel that is opened when the receptor is bound by an agonist. A metabotropic receptor is a receptor protein that doesn't contain an ion channel, but when activated may use a G protein system to alter the functioning of the postsynaptic cell. A receptor subtype is any type of receptor having functional characteristics that distinguish it from other types of receptors for the same neurotransmitter. A G protein coupled receptor, or GPCR, is a cell surface receptor that when activated extracellularly initiates G protein signaling mechanisms inside the cell. Any substance that binds to a receptor is a ligand and has several kinds of effects. A ligand that is classified as an agonist, a molecule, usually a drug, that binds a receptor molecule and initiates a response like that of another molecule, usually a neurotransmitter, initiates the normal effects of the transmitter on the receptor. A receptor antagonist or molecule, usually a drug that interferes with or prevents the action of a transmitter, is a ligand that binds to a receptor and does not activate it, thereby blocking it from being activated by other ligands, including the native neurotransmitter. And an inverse agonist is a substance that binds to a receptor and causes it to do the opposite of what the naturally occurring transmitter does. It's a less time and type of ligand that binds to the receptor and initiates an effect that is the reverse of the normal function of the receptor. So drugs with any of these three actions are properly called competitive ligands, or substances that directly competes with the endogenous ligand for the same binding site on a receptor molecule. The non-competitive ligand, also known as the neuromodulator, is a substance that alters the response to an endogenous ligand without interacting with the endogenous ligand's recognition site. A modulatory site is a portion of a receptor that, when bound by a compound, alters the receptor's response to its transmitter. So many chemical neurotransmitters have been identified. To be considered a neurotransmitter, a substance should: the substance exists in presynaptic axon terminals. The presynaptic cell contains appropriate enzymes for synthesizing the substance. The substance is released in significant quantities when action potentials reach the terminals. Specific receptors that recognize the released substance exist on the postsynaptic membrane. Experimental application of the substance produces changes in the postsynaptic cells, and blocking release of the substance prevents presynaptic activity from affecting the postsynaptic cell. The different types of neurotransmitters include an amine neurotransmitter, which is based on modification of a single amino acid. Examples include acetylcholine, serotonin, and dopamine. An amino acid neurotransmitter is an amino acid. These examples include GABA, glycine, and glutamate. A peptide neurotransmitter, also known as a neuropeptide, consists of a short chain of amino acid. These examples include neuropeptide Y, galanin, and VIP. And gas neurotransmitters, which are soluble gases such as nitric acid or carbon monoxide that is produced and released by a neuron to alter the functioning of another neuron. So the neurotransmitter systems form a complex array in the brain. Colocalization, also known as co-release, is the appearance of more than one neurotransmitter in a given presynaptic terminal. The most abundant excitatory and inhibitory neurotransmitters in the brain are amino acids. The most plentiful excitatory neurotransmitters in the brain are glutamate, the most common excitatory transmitter, and aspartate, which is excitatory at many synapses. There are two families of acetylcholine receptors in the peripheral nervous system and the central nervous system. Nicotinic, which is cholinergic receptors that respond to nicotine as well as acetylcholine, and muscarinic, which is cholinergic receptors that respond to the chemical muscarine as well as to acetylcholine. The mesostriatal pathway originates from the midbrain, specifically the substantia nigra, which literally means black substance. The mesolimbocortical pathway also originates in the midbrain in the ventral tegmental area, or the VTA, which is a portion of the midbrain that projects dopaminergic fibers to the nucleus accumbens and projects to the limbic system and cortex. And finally, some neurotransmitters are gases. They include nitric oxide, which is a soluble gas that serves as a retrograde gas neurotransmitter in the nervous system, and a retrograde transmitter diffuses from the postsynaptic neuron back to the presynaptic neuron.